Hi dear students. Today we are going to discuss about mitosis, the type of cell division in which number of chromosomes remain constant in the daughter cells. And the parent cell is called mitosis. Mitosis can take place in the haploid and diploid cells in all parts of the body. It is divided into two phases, karyokinesis and cytokinesis. The division of nucleus is called karyokinesis, while the division of the whole cell is called cytokinesis. Now we will discuss the subdivisions of karyokinesis. Prophase pro mean first and phase mean stage. The chromosomes are not visible even with electron microscope during interphase of the cell cycle. The chromatin material is condensed by folding. Thus the chromosomes appear as thin threads. 1. The nuclear envelope disappears. 2. Nucleoli disappears. 3. Mitotic apparatus is organized. 4. Cytoplasm becomes more viscous. 5. Chromosomes become thick. Metaphase. The chromatids are attached at a point called centromere. The centromere has special areas called kinetochore. Kinetochore fibers of spindle attached at the kinetochore of the chromosome. These fibers align the chromosomes at the equator of the spindle. Thus they form metaphase plate in the equator. Each kinetochore is attached with two fibers each from opposite poles. Anaphase. It is the most critical phase of the mitosis and shows equal distribution of chromatids in the daughter cells. The kinetochore fibers of spindle contract towards their respective poles. At the same time, polar microtubules elongated. Telophase. It is the last phase of karyokinesis. The anaphase ends with reaching of the chromosomes at positive poles and telophase starts. The chromosomes unfold in decadence. They ultimately disappear as chromatin. Mitotic apparatus disorganized. The nuclear membrane and nucleus reorganize, plus two nuclei are formed at the two poles of the cell.
Please subscribe to my channel Scientific World.